Chapter 11 concerns controlling the growth of microorganisms in the environment. And so we'll do this in two ways, either by altering the environment physically, by changing the temperature, the moisture, or the pressure, or by treating the environment with chemicals that will affect the microbes' ability to grow in some way. So this is important in a lot of areas like restaurants, your own home, especially so in the image shown on this slide of an operating room where we try to reduce the growth of microbes as much as possible to eliminate the possibility of infection in the patient. Lots of terminology in Chapter 11. So these terms listed here are often used. Some of them are misused. So things to pay attention to when you're reading through these definitions is how much of the microbial life is destroyed. So for example, in sterilization, the goal is to kill all microbial life, whereas the others, the goal is to kill most microbial life. Also pay attention to where the microbial life is being controlled. So it could be on an inanimate surface, like something non-living, as in disinfection or decontamination, sanitization, or it could be on a living surface like a patient's skin, as in antisepsis degerming, or also in decontamination, sanitization. And in decontamination sanitization, mechanical removal is essentially scrubbing. So you're scrubbing the surface or the skin in order to physically, mechanically remove microbes from that surface. With the different groups of microorganisms, there are varying levels of resistance. So in this figure from your book, we have put in a hierarchical fashion the least resistant at the bottom. So these are the microbe groups that are going to be the easiest to kill or to deal with in the environment by using chemical or physical methods. And then at the top of this figure are the most resistant groups of microbes. So these are the ones that are going to be very challenging or very difficult to kill. So I'd just like to point out things that we've learned about in this class. We have enveloped viruses and non-enveloped viruses. So those are both relatively easy to kill or deal with through chemical or physical means. Gram-positive and gram-negative bacteria. We have the gram-negative bacteria being more difficult to kill, mostly because of that double membrane and the cell wall. So there's kind of three layers to penetrate. Where in the gram-positive bacteria, we have only the inner cell membrane and the cell wall, so just the two layers to penetrate. And then we have briefly discussed endospores, which are bacteria that have the ability to form spores if the environmental conditions are not favorable. And so endospores are really resistant to dehydration, chemical treatment, heat treatment, radiation treatment. So endospores are really, really hard to kill. And we haven't talked about them in this class, but prions, which are misfolded proteins and can cause Creutzfeldt-Jacobs disease and mad cow disease in animals, are among the hardest to kill. So there are no great ways to kill prions. So these are a factor in a lot of the neural surgeries where you're dealing with nervous tissue where prions could be present. And then looking at, in this chart, we have comparison of endospores. So these are bacteria that have the ability to form spores when conditions are not favorable. And then we have that compared to vegetative forms. So vegetative form of cells are those that are living, actively dividing, and actively metabolizing, where the endospore is not metabolically active and is essentially a protein coat containing DNA only. So if you look at different methods of controlling microbial growth, including heat, radiation, a sterilizing gas, which you just spray the gas on the microbe and it would kill the microbe, or sporicidal liquids like formaldehyde and glutaraldehyde. If you compare the endospore required treatment to the vegetative spore required treatment, we see in all cases that the endospore is harder to kill than the vegetative cell. And that agrees with this past figure where the bacterial endospore is among the most resistant of all groups, and all the living cells that are below are going to be a lot easier to kill. So with heat, you have to apply more heat to an endospore. We're looking at 120 degrees centigrade. You have to apply more radiation, and the unit there is grays. We have to apply higher concentration of sterilizing gas, and you have to allow a longer time for soaking with sporicidal liquids with endospores versus vegetative forms. With all of these control methods, we're going to refer to them as either being bactericidal, which is when they kill bacteria, or being bacteriostatic, which is when they just inhibit the growth of bacteria, but they do not kill the bacteria. In related terms to these, you can replace the beginning of the word with microbio, germa, or microbio. So microbiocide would be any chemical or treatment that kills microbes. Germicide would be anything that kills germs. Microbiostatic would be any chemical or treatment that prevents the growth of microbes. So you can mix these terms up, but the ending of the word tells you if it kills or just stops growth. And then in addition to whether the bacteria, what type of bacteria or microbe you're dealing with, if it's an endospore or a vegetative cell, there are many other factors that affect how quickly microbes die in the presence of different treatments for controlling their growth. So the first graph in part A is time. So if you incubate a microbe with a chemical or with some sort of treatment for longer, you will kill more of the cells. So time is a factor. The number of microbes. So the top line in the graph in part B represents a high load, meaning a lot of microbes are present when you start the treatment. And the lower blue line represents a low load, so there are less microbes present. If you have a high load of microbes, it's going to take longer to sterilize, which means killing all of the microbes present, than if you had a low load. So the number of microbes will affect how quickly microbes die. The type of microbe, which we already mentioned, so spores are going to be harder to kill and take longer than vegetative cells. And then the type of agent that you use, so depending on the chemical or the treatment, if it's microbiostatic, then you're not going to reduce the number of cells because that will just stop it from growing, but those already there will remain. If you're using a microbiocidal treatment, then you will see a reduction in the number of microbes as you leave the treatment on the cells. Okay, so all of these techniques, which we're going to group them into either physical methods of microbial control or chemical methods of microbial control. And we're going to talk about the physical first. But both physical and chemical methods of microbial control are going to affect the cells in 4 ways. So the first thing that they may do is they may damage the cell wall. So in bacteria or fungi, we have a cell wall present. And a lot of these chemicals or environmental manipulations will affect the integrity of the cell wall so that it essentially breaks down. The second way that these methods of control can target the microbe cell is by affecting the cell or cytoplasmic membrane, and that's what's shown in the image from your book here. So surfactants is one example, and soaps are a type of surfactant. So what the soap will do is it will wedge in between the lipids of the cytoplasmic membrane, and it will disrupt it, so then the contents of the cell leak out and the cell will die. We have some methods will affect the synthesis of DNA or RNA. So we have either they prevent DNA or RNA from being made, or the DNA or RNA is damaged, as shown in this image from your book. So radiation is an example of a control method that affects cells in this way. It causes DNA to break, or it causes abnormal bonds in the DNA. And that would be really bad for the cell because it would now not have all the instructions it needs to do all the essential jobs. And then synthesis of proteins is the last way that physical and chemical control methods affect microbes. So we have a protein in its native state, which is shown in part A. So this is how the protein normally should be folded up inside the cell. So some of these control methods will cause complete denaturation, which is when the protein just completely unfolds and has a very, very altered shape. We have some of these methods will slightly alter the shape of the protein, or we have some methods that may block the active site of the protein. So especially if it's functioning as an enzyme, can't bind to the substrate anymore, and then that would be bad for the cell because that chemical reaction would not be catalyzed. So either affecting proteins being made or the way proteins function is the last cellular target. Okay, so let's do the physical control methods of microbial control. So we have first heat. So heat will kill microbes, so it's bactericidal. It kills bacteria or microbes by denaturing proteins and causing them to unfold. And so each microorganism has a characteristic thermal death point, which is defined as the lowest temperature at which all cells in a culture are killed. And they also all have a thermal death time that is characteristic. So at a given temperature, the time it takes to kill all the cells in a culture. Moist heat is more effective than simple dry heat. So in moist heat, you have moisture plus heat. And in an autoclave, you have pressure added as well. And these three things together, moisture, heat, and pressure, are incredibly efficient at denaturing proteins and bactericidal, so killing the bacteria. Pasteurization is the application of mild heat that will reduce the microbe count, but it will not damage the product. So although heat is bactericidal, if you use heat in pasteurization, you do not heat until all microbes are killed, so some remain. And there are different ways of doing pasteurization. It's pretty much exclusively used in food so that you don't change the taste of the food. So in dairy, for example, you can have high temperature for a short time. So this is what's done with refrigerated milk. There's also ultra high temperature sterilization, which is what's done with dairy products that don't have to be refrigerated. And you do not need to memorize these temperatures and time points. Just know that pasteurization is the application of heat, but it does not achieve sterility. Desiccation, which is just drying things out, and cold temperatures will both suspend microbial metabolism, so they basically shut the cell down, but they do not kill the cell. So when you heat back to normal temperature or you rehydrate, then the microbes present in the food or the nuts and lentils shown in these images will then be able to divide and will contaminate the food or continue to contaminate the food. So these two methods are bacteriostatic. Radiation, which is shining high energy wavelengths. So these are UV, X-rays, and gamma rays onto things, is bactericidal because it causes damage in the DNA. And one example of that damage is shown here, where we have a thymine dimer. So in the presence of radiation, two thymines next to each other on a DNA molecule will bind to each other and cause a bend in the DNA molecule that will be problematic for the cell. We use irradiation in our food supply. By shining radiation on foods, we can reduce the microbe count and kill any microbes contaminating the food. Filtration is when you pass a liquid through a membrane with very small pore size. So this can remove most microbes. Viruses can often get through, but bigger microbes like bacteria or fungi will be eliminated from the liquid through filtration. And then high salt or sugar, which is what we do when we preserve fruits or vegetables in jam and jelly, or country ham, giving high salt to the meat so that any microbes present in the meat will undergo plasmolysis, where the water will leave the cell in the presence of high salt or sugar, and this will cause the cell to shrivel. The cell wall remains with the same structure, but the cell membrane will shrivel and the microbe will die. So this is bactericidal. So this summarizes all of those. It tells you the effect on the microbe and then whether they're bacteriostatic or bactericidal. So these are all the physical methods of microbial control. The chemical methods are just a list of chemicals. So I'll go through these pretty quickly because most of the information you need on these is listed on the slides. Okay, so the first are the halogens, so chlorine and iodine. So this is used in hospitals, home disinfection. Pretty much every household has a bottle of bleach. Food processing. So we also wash eggs, for example, the shells in a diluted bleach solution. These are great chemicals for reducing microbe growth because they are sporicidal, so they can kill spores. However, they are inactivated by organics, meaning if there's a lot of organic matter like blood or feces or urine, then that can inactivate the chemical, so it doesn't work in the presence of those. Phenol was one of the first disinfectants or sterilants. It's no longer used because it can be absorbed by the skin and will cause nerve damage. So there's kind of a derivative now of phenolics. The things that all phenols and phenolics share is the six-sided ring structure. So this is called a benzene ring. It's present in each of these compounds, which are in this category. You do not have to know the chemical structure of these compounds. I just wanted you to know that's what they all share, is that part of their structure. So the way they work is they denature proteins and they destroy cytoplasmic membranes. They're really good in hospitals because they are active in the presence of organic compounds like lots of blood. They're really stable for a long time and they're very persistent. Alcohols are very commonly used, and the point I'll show you from this table that's included is that at 100% concentration, alcohols will allow growth. So in this table, G stands for growth. So no matter what the time of exposure, at 100% concentration, we have growth of microorganisms in the presence of this concentration of alcohol. So alcohol must be diluted with water in order to be effective. So the concentration most commonly used is 70%. So you can see at 70% alcohol, 30% water mixture, we have no growth of microbes at any of the times of exposure. Hydrogen peroxide is usually used as a 3% solution. So it is sporicidal and able to kill bacteria that can form endospores. It works well in organic matter and skin wounds, and it's also really commonly used in contact lenses. So hydrogen peroxide creates a lot of free radicals, which are molecules that are very reactive, so they'll react with components in the cell, and in that way will destroy the microbe. Surface active agents are soaps, detergents, and quaternary compounds, abbreviated quats. So these are all types of surfactants, and what they do is they lower the surface tension to mechanically remove microbes from the skin. So if you have microbes stuck to your skin, you soap up your skin, the microbes will get caught in the soap bubbles, and when you wash the bubbles with water, the microbes will be washed off your skin as well. Heavy metals are found to have antimicrobial properties by inactivating microbial proteins. The only ones still used are mercury and silver, and they are sometimes incorporated into hard surfaces like the floor of a hospital or a hospital table. Aldehydes are the most commonly used disinfectants in hospitals, so these include formaldehyde and glutaraldehyde. So they are desirable in hospitals because they're sporicidal and they are not inactivated by organic matter. They are, however, not used on any living surface because they're toxic and irritating. They do often use them for sterilizing hospital instruments like the endoscope shown on the left and the respiratory machine shown on the right. And that concludes Chapter 11 material.